Hello students, welcome to today's lesson on genetic engineering. In this lesson, we are going to study genetic engineering in bacteria and genetic engineering in plants. What is genetic engineering? Genetic engineering is the process of transferring genes from one organism to another organism without species barrier. This process discovered and designed to produce desired products to satisfy unlimited human requirements. In genetic engineering, to transfer genes from donor organism to recipient organism, it's possible to transfer either the donor and recipient organism belongs to the same species or different species. Why we mostly use bacteria to produce desired products? This is because bacteria gives us high amount of desired product in short period of time, since bacteria have short life cycle or short reproduction period. The development of three main techniques make genetic engineering possible. So, what are these three main techniques? The first technique must be cut gene of interest out of the DNA molecule by using restricted endonuclease enzyme, which act as a molecular scissor. The second technique must be insert or tie gene of interest into another DNA molecule, mostly the vector DNA molecule, through the action of ligase enzyme. The third technique must be transfer gene of interest into another cells by using vector. Vector means a medium or a means of transferring something. Vectors in genetic engineering can be viruses like lambdaphage and bacterial plasmids. These are the two fundamental vectors that most scientists have been using to transfer genes from donor organism to recipient organism. Let us see genetic engineering diagrammatically. From the first diagram, as you watch, plasmids which will act as vectors should be isolated from bacteria. Second, this isolated plasmid should be cut open by restricted endonuclease enzyme. Third, gene of interest could be cut out from donor DNA in donor cells by using the same restricted endonuclease enzyme what we applied to cut open plasmids. Applying this same enzyme is very important to form sticky ends on both plasmids and gene of interest. Fourth, open up plasmids and gene of interest should be ligated or tied by using ligase enzyme, and hence this combination forms recombinant plasmids. Fifth, both recombinant plasmids and bacteria should be incubated in laboratory on petri dish. In this process or in this way, bacteria will take up the plasmids, recombinant plasmids. Then we should culture these bacteria and let them reproduce in laboratory until the mass or number becomes high. When their number becomes high, we should shift them to seed tank and then to fermentation tank to produce the required amount of the product of gene of interest, what we discussed in the last class, antibiotics production. Because these genetically modified bacteria have been used to produce a range of products like antibiotics, vaccine, enzyme, hormones, and etc. Genetic engineering in plants. Many crop plants have also had foreign genes inserted into them for different purposes or reasons. This main reason is that inserting foreign genes into the plants mostly intended to produce new properties. These new properties that plants can acquire due to genetic engineering are resistance to infectious disease, resistance to animal pests, resistance to weeds, a longer shelf life before decaying, grow in different agroecology, and drought resistance. So, by inserting foreign genes into plants, we redesign them to give us high yields. Genetic engineering in plants is not as simple as genetic engineering in bacteria. Genetic engineering of plants posed problems for biologists because plants will not accept plasmids as bacteria cells do accept. So, scientists to genetically modify bacteria, they rely on Agrobacterium tumefaciens that regularly infect plant cells. So, biologists use this Agrobacterium as a vector to carry genes that have to be inserted into the bacteria and then transferred into plants. However, Agrobacterium can't be used to genetically modify all types of plants. So, gene gain shoots gene of interest into cells of plants by using a tiny pellets of gold as bullets that are covered in DNA, what we sometimes call golden gene. The gene gain has made it possible to genetically modify plants such as maize, tobacco, carrots, soybean, and apple. These are the 5 plant species that cannot be infected by Agrobacterium tumefaciens. So in this case, we need to have a gene gain to genetically modify plants like this. Here are the techniques how we can genetically modify plant species. First, DNA should be cut with restricted endonuclease enzyme to isolate gene of interest from the donor species. Second, plasmids must be isolated from Agrobacterium tumefaciens. Then, this plasmid must be cut open with restricted endonuclease enzyme. And third, we have to link the cut plasmids with desired gene or gene of interest. After then, we formulate recombinant plasmid DNA. Then, we have to incubate this recombinant plasmid with leaf discs on Petri dish for a certain period of time. Leaf discs floated on the liquid containing the plasmids. Some will take up the plasmids. Then leaf discs should be cultivated on a nutrient medium, what we call micropropagation. Then when the leaf discs started to formulate roots and shoots, after a certain periods of later, the leaf discs gradually developed to plantlet. Then these plantlets grown into whole plants whose cells now contain the foreign gene. These large plantlets must be transferred to and planted on the ground.